right, good morning, everybody. Thanks for joining us this morning. We've got our first in the fall 2021 MSSE Industrial Seminar Series this morning. We'll have actually two U of M faculty join us today. Dan Boley will join us a little bit later and talk a little bit about common machine learning methods, and he'll give kind of a pictorial overview of how some of those work using some examples. But first, we obviously have Dan Knights with us, and his talk is entitled Getting More Signal with Less Data. Dan, go ahead.
All right. Well, nice to be here with everyone. I'm really impressed with your commitment. It's normally like iPad time for my kids, and I'm usually lounging around in bed right now. So I kept a little bit of my bed hair coming into this. I thought it would lighten the mood a little bit. So I'm going to talk about some work that's been going on. I try to kind of cover like the general arc of stuff I've been doing in my lab, but then like zoom in on some computational methods too, so that you get like a little bit of both. And I can give just like a little more. Here, let me share my screen. A little more introduction to myself and my work. So that's me. I'm in the computer science department and in the biotech institute at the U of M. So it's a little bit of a like I'm like sort of halfway between computer science and biology. So we call that computational biology or bioinformatics. Disclosures, I serve as a paid senior scientific advisor to Diversigen. And this is a company that I'll talk about a little bit at the end. It's a company that I started a few years ago, because I know some of you are in, working in industry, and maybe there'll be time for questions at the end. Maybe it's something you want to chat about too. So my lab is working on computational methods for genome and metagenome data, and also, more generally, just kind of modeling the natural world. And I'm also like a puzzle enthusiast, like to do, Rubik's Cube and all of those sort of sequential movement puzzles. I actually, my wife and I have a cabin up north, and we were setting up we were trying to figure out how to fit 4 kids in one room without having like bunk beds, because we wanted to be able to use the room for other stuff. And so we figured out we could put 2 day beds, and a day bed is like a thing that looks like a couch. But then you can also sleep on it, and then it has a trundle that pulls out underneath, so you can have like 2 beds for the price of 1 couch. And we figured out we could fit the day beds, and that if you pulled one of the trundles out and rotated it and then slid it, then you could pull the other trundle out and fit both trundles in this one room. And I realized it was like a sequential movement puzzle. Like my friends were up there and they were texting me saying like, it doesn't fit. Like they couldn't figure out how to pull out both of the trundles, because you had to like go through this series of steps to make them fit. So I guess that sort of carries over in other parts of my life. And I really like rock climbing too. You can see this picture of my lab from a few years ago on one of our climbing trips. I'm going to be talking today about some of our work that's focused on the microbiome, which is it's been in the news. I see it in the news all the time, because I'm looking for it, but I'm sure some of you have seen it. Like fecal transplants have been this miracle cure that people have made jokes about. It was like in a South Park episode where you actually like transplant stool from a healthy donor into someone with an infection. But there's all these other applications. And this is a picture of your microbiome. It's an electron scanning micrograph of a gut sample, meaning a stool sample, zooming in on the microbes, and then an artist colored it to make it look nice afterward. So good morning to you. And this is the point of this is to show how diverse it is. It's really complex in there. It's like a whole ecosystem. And that's why we use genomics, and we need these like sort of high dimensional computational methods to study the microbiome. And the microbiome is very interesting right now in medical fields because it's linked to all these diseases like diabetes and obesity. So anything like metabolic has a microbiome signature. You know, you have a metabolic disease, your microbiome is also a different ecosystem. Autoimmune disorders, Crohn's disease, ulcerative colitis, rheumatoid arthritis. These are all linked to having a different ecosystem inside you. So it's sort of like climate change, but it's inside in your gut. And you can see, you know, like a lot of these diseases are also rising as the world becomes more industrialized. So that's a correlation, but maybe there's something, maybe there's a real causal relationship there. So these are two diseases that are going up all over the world, and they're especially high in industrialized countries. And it turns out that when you move from a non-industrialized country to an industrialized country, you also acquire the risk of those diseases within your own lifetime. So it's not like a genetic thing, or it's not, you know, the human genes at least. This is something, these are just some data from some groups of US immigrants. This is some data that we collected in my lab showing body mass index going up, the fraction of people with obesity who've been here for less than 1 year versus 15 plus years. And then this is people who were US born in the same city. So you can see that people are sort of acquiring the US risk the longer they stay in the US. And this is in kids, this is just over 3 years, you see the incidence of obesity and being overweight is like doubling. So we thought this was kind of interesting, and we were interested in understanding, like, is the microbiome related to these changes and this risk? And I'm going to use this as a case study to kind of show you what the data are like, the kinds of inferences we can make, and then to show you why we needed to develop some new computational methods. So this is chapter 1, born in the USA. This is, or I should call it not born in the USA. This is a study that was run by Pajao Vange, who was a bioinformatics student in my lab. And she is ethnically Hmong, and there's a large Hmong population in the Twin Cities. So a lot of the work we did was with the Hmong. And what we're trying to answer was what happens when you move from here, which is a refugee camp in northern Thailand, to urban Minnesota. And to do this, we had to get samples from people who were still living in Thailand. These are 2 different refugee camps we went to. We also worked with the Hmong. We also worked with the Karen, who are an ethnic group that were fleeing Myanmar because of ethnic persecution. And then, so we got samples from people still living in Thailand. We got samples from people who are living in the US. And we also have samples from 2nd generation in the US. So these are people who were born in the US, but their parents weren't, but still from the same ethnic group. And then we have US controls, which are like European Americans who, have been in the country for multiple generations. This is what we're actually doing kind of on the back end to make the study happen. So we collect lots of stool samples from these individuals, and then we do DNA sequencing on it. We can't go in and like try to grow all the bugs because they're very finicky and they don't grow easily in the lab, and that would take forever. We treat it just like the human genome. We get the bacterial DNA out and run DNA sequencing. And so we get like a giant file of millions of DNA sequences for each subject, for each time point. And then we compare those to a reference database. And this is going to come up later when we dive into some computational methods. How do you actually do that comparison? It's a very, very like fundamental problem in computer science and in bioinformatics. It's sort of like, string search, string matching. And once we do that, we get this table of, it's just, like a standard data table of every subject. You have all these features, which are the strains that they had, the bacterial strains or species. And you learn what fraction of each, what fraction of the gut was each strain. We can take that table and do all sorts of other things like figure out the distance between every subject and then project that into a few dimensions so that we can visualize the data and see if there are any trends. So this is often like the first thing that we do is just sort of project the distances down and try to look for any kind of clustering. And that's what we did with these data. And there was some really obvious trends. When we first looked at the data, it was like a giant blob. But then when we colored it by the different immigration groups and ethnic groups, it was pretty obvious what was going on. So these two circles on the left are encircling like one standard deviation or two standard deviations of the, there's a 95% confidence interval of the two ethnic groups still in Thailand. So the green are the Karen and the pink are the Hmong. This gray circle is the US controls. Everyone in the background is first generation immigrants from these two groups. So we saw, basically like a range of people sort of spanning between the pre and post immigration when they were first generation. And then this final circle on the right is the second generation. So basically by the time people are in the US for a whole generation, their microbiomes were like pretty much overlapping with the US controls. And this was really a group effort doing this study, 550 subjects. And we did it using community-based participatory action research methods, which means people from these groups were actually involved in carrying out the study. We had a community advisory board who helped us understand what would be like cultural sensitivities when we're carrying this out. What was the community interested in learning about their own data? How should we disseminate this? And then community researchers were paid to help, recruit subjects and so on. And that's where some of the questions came out too that we'll look at in a minute. The other thing we can do with these tables is figure out the diversity. So like a really naive diversity index would be just how many strains do you have? What's the total species count in your gut ecosystem? And like in this example, subject 1 has 4 unique species, subject 2 only has 2 unique species. So we did that on all these samples. And this was kind of like the real like jaw-dropping thing in the study. So I'm going to show you the diversity of the people living in Thailand. And this is actually phylogenetic diversity. It's a little bit more complicated than the one I showed you. If you can learn about all of these types of methods in the class computational genomics, which I normally teach in the fall. I think Ray Kwong is teaching it right now. And so that you just think of this as a relative measure for now. So there's this range of diversity in people living in Thailand. And when we did the study, one of the questions that the community was interested in was obesity. We heard anecdotally from someone who said, oh yeah, we were in the refugee camp and you find out that someone is like going to the USA, and someone might tell them like, oh, that's awesome, congratulations for you, but you're going to get fat. And they wanted to know, like, is the change in the microbiome in the ecosystem in the gut related to risk of obesity? So when we did the study, we carried out in each group, we stratified and made sure that we got lean individuals and individuals with obesity in each category. And what I'm showing you here is just in Thailand, this is the diversity, the difference in diversity between lean and obese. And so you can see what's happening here. People with obesity have a less diverse ecosystem in their microbiome, still in Thailand, living in Thailand. Now, the next group is first generation, and you can see diversity has gone down again. And it's actually almost or just about twice the effect of obesity. So these are lean individuals living in the US who were born in Laos or Thailand or Burma. And their diversity has gone down twice as much as the people still living in Thailand who have obesity. Now I'll show you first generation obese. Can you guess where it's going to go? So now there's like this compounding effect. In living in an industrialized country seems to, this is a correlation, so we haven't demonstrated causality, but it implies that it's causing this, big drop in diversity. If you live in an industrialized country and you have obesity, it goes down again. Then we have second generation obese, and then US controls. And again, by the end of, 2 generations, it seems that you kind of end up in the same boat as people who've been living in the US for many generations. And this is the first time there's this theory about missing microbes that of this sort of like diminishing diversity from generation to generation in our microbial ecosystems, because the parents get antibiotics, kids are born through C-section, kids are fed formula, maybe there's other types of like we're not eating enough fiber or whatever. So in one generation you kind of lose some diversity, and then you can't pass it on to the next generation. So they start at a lower baseline, and then they lose some of theirs for the same reasons, and then pass on fewer to their kids, and so on. And so there's some scientists who've been like sounding the alarm, saying we really need to be like watching this because we evolved with these microbes. We don't want to have them all disappear. And this is the first time that we had actually seen it happening across generations. So we wanted to understand more like what's going on and what species are changing, what are they doing, what enzymes do they have, why is this happening? It turns out all these, and this is where we'll get into some of the more computational stuff, all of these data were collected with this very low resolution approach where we just sequence one marker gene. It's cheap, but it doesn't really help tell you what's going on like functionally. We can do a much higher resolution assay. And in fact, in this study, we had funding for it and we did that. So we did a whole genome shotgun sequencing on a subset of about 10% of the subjects. And this costs like 5 to 10 times more than the low resolution approach. And one of the things we were able to see, so here I'm showing you, these are Hmong people living in Thailand. So every column is a subject. the rows are different enzymes. Enzymes are proteins that like break down parts of your food, basically. And the colors just show, do they have a lot of that enzyme, which is purple, or a little, or very little, which is green. So you can see in the Thai group, there's a lot of whatever enzymes these are, and not a lot of the ones at the top. And then these are US controls. Here you see there's a lot of these other enzymes and not a lot of those. And then what do you think the first generation are going to look like? These are going to be people who were not born in the US, but have been living in the US for 10 years or more. So it turned out, when we looked with this higher resolution method, we saw almost all of these enzymes that were present in the ecosystem in Thailand, the gut ecosystem in Thailand, were very low abundance, and some of them had gone like completely missing, or almost completely missing. And this gave us a little hint of what was going on. So some of these ones that had gone missing were related to different types of dietary fiber, and some of them break down fibers that are in plants that are commonly grown and eaten in Southeast Asia, like palm, coconut, konjac, tamarind. And so that might explain part of the shift is, you stop eating certain types of plant fibers, the bugs who relied on those are just gonna like go away. And it's not necessarily a bad thing. And again, this is all, there's nothing causal here. This is just an observation. But what we did observe is that loss in diversity. So it could be that, yeah, when you move countries, you start losing your native microbes and you start picking up the microbes in the new country. But it seems that at least when you go from non-industrial or recently industrialized to a fully industrialized country, the new microbes you pick up don't really make up for the ones that you lose. So as I mentioned, this is like the primo data, but it's very expensive, very high information content. So I'll call that WGS for whole genome shotgun sequencing. The other kind is called 16S. That's just the name of the marker gene, the one gene that we were sequencing. The 16S approach costs like $50 or so, maybe a little less. The shotgun sequencing costs 500. And ideally we would run the shotgun on every study, but that would break the bank. We're limited by grant funding and things like that. So to me, it seemed like we really needed some methods here. What can we do that's lower cost but still high resolution? And that's why, in this particular study, we had done 16S on all 550 samples, the WGS only on 55. I want to be able to do high resolution sequencing on everything in a study of 500 samples. So there were two key steps in my mind. One was, let's try to get more signal from the data. Maybe we're being greedy with the deep sequencing. Maybe we're just throwing money at it and paying for more sequences, but we're not being very parsimonious with our consumption of the data. Maybe there's more information content in there that's being wasted. And then I worked with some people on the wet lab side who could help get the costs down on kind of the sample prep and things like that. So I'm going to try to decrease the amount of data we need. And then, so you don't have to pay as much for the actual DNA sequencing. And then my partners are going to try to decrease the cost of like getting the samples ready for the sequencing. And if we can do both of those, then the whole cost will go down quite a bit. So this brings us to chapter 2, fast and spurious. Remember, I said we learn about these data by mapping the input DNA to the reference database. And what basically everyone in the field was doing was using approximate search tools. And the reason for that is that it's really slow to do optimal alignment. But when you use these approximate tools, they make a lot of mistakes. They'll often find the wrong match, or a close match, but not the best one, or not even find a match when there is one. And there is actually an optimal solution for this. It's called Needleman-Wunsch alignment, and it uses dynamic programming. So this is something that it's like one of the first lectures, or one of the first topics you go through in a computational biology class. Basically, you take the rows as the characters in the search string, the query. The columns are the characters in one reference string. And then you kind of fill the table in row by row, making sure that the current choice is optimal given the choices around it. And it turns out by the time you get to the whole table filled in, you can trace back and find what's guaranteed to be the optimal solution, because any particular part of that, the whole optimal solution, also has to be optimal. So it's got that kind of like optimal decomposition feature that you need to have for dynamic programming to work. And so what this does is make the whole problem sort of tractable. It takes it from being like highly exponential to just m times n operations, where you have to fill in the whole table. So can we just use this? Why not just use this every time we get the data, and maybe we'll get more information out? The problem is, in a typical data set, we might have, say, a total of 10 billion query sequences. These are the DNA sequences that we paid for, that we got with the DNA sequencing machine. Those might be 100 bases long. Multiply those together to figure out how many query cells we have, times 10,000 reference sequences in the database or more. So those are all the reference genomes of the known strains of bacteria, times those bacterial genomes are not 100 bases long. They're like 5 million bases long. And so in total, you get 5 times 10 to the 23 cells, which is 1,000 times more complex than all of the possible states in Rubik's Cube, which is 43 times 10 to the 19. And so this would take forever to run. If you wanted to find approximate compute time for a single shotgun, deep whole genome shotgun sequencing sample, using the Needleman-Wunsch optimal algorithm that was published in the 70s, it would take a supercomputer with 100 nodes about 4 years of compute time. So if you did that, across 500 samples in a study, now you're looking at 2,000 years of compute time to get optimal results. Over the subsequent couple of decades, there were groups that worked hard on parallelizing this and speeding it up in different ways, and got the cost down by like a factor of 20, which is pretty cool. So it's now 75 days, but that's still too long. If you're doing that on 500 samples, like that's just too much compute time. So there was a bright PhD student in my lab, Gabe Algaleith, who was working on this problem. And this is one of those things, like, he kind of stumbled into this because he was trying to make a better approximate aligner, like a better heuristic search tool. And in order to evaluate it, he wanted to quickly whip up a faster optimal search tool so that he could get like the gold standard. And we were talking about this and came up with this idea that, well, as you get toward the bottom of this table, you're kind of filling it in row by row. there must be parts of the table where you could prove that even if the rest of the alignment starting at that point was perfect, you still couldn't do better than proceeding from another part of the table. So normally in the algorithm, you can only trace back once you've finished, but at a certain point, you can look forward a little bit and put some bounds around, like, what's the best and worst possible score you could get descending from a particular cell. And if you do that, you should be able to prune the table a little bit. So Gabe tried that, and it's pretty cool. This was actually, I did find this in someone's thesis like 10 years ago. So this has been done before. And it worked. It cut like half of the cells out of the table on average. So I'm going to pause. That this is the first time we're getting into any like computational stuff. So I'm going to pause and see if anyone has questions about this, or like general questions about the previous study that I talked about.
Does this have a way that it kind of ends up relating to the point jump algorithm for path planning in a more general sense, I guess you'd say?
I don't know that particular algorithm. Is it also solved with dynamic programming?
I don't remember off the top of my head. I remember some of the vagaries about it, that it cuts out a lot of the cells on a two-dimensional map to, like, if you don't run into an obstacle, you just keep going further instead of trying to replot your path.
Yeah, it might. I think it's really good to think about ways in which this might be analogous to things in other fields, because what's about to happen is pretty cool. Like, I'm setting up a joke, or like a story, and then we haven't gotten to the punchline yet. So if you can find an algorithm that's like this, you could port over what Gabe did, that I'll show you in a minute, to that other algorithm in another field. Other questions?
Yeah, so a 1/2 reduction of the complexity, is not obviously going to be a game changer. So what gave you guys sort of the intuition that this would be fruitful? Because like if you can solve half of the problem, you could probably solve the whole problem. So reducing it by half, it isn't obvious that is going to like really open some doors for you. So, did you guys just sort of have a gut feeling, or you're like, well, this is what we got?
Yeah, that's a good question. We didn't think we were going to solve the whole problem, and we were just kind of poking around and thought like, hey, this is pretty easy. Let's just try this out, see if we can, because even like a 2x speed up might be nice for building test cases. So I think if we had started with the goal of solving the whole problem, we might never have even started working on it. Other questions?
I got a question on the study that you had.
Yeah.
Since the microbiome, whatever, I don't know what it's called, but the bacteria, or I think what it was in the people that were living in Thailand, was higher. Was the diet less like processed foods, like prepackaged processed foods, and more like would that play a factor, or maybe like their farming principles, or like maybe they don't use as many like pesticides as we do?
Yeah, those are actually both hypotheses that we're not following up on the farming pesticides one. But the processed foods, I think, is very interesting, and could be, one of my theories is that it's all of the like preservatives in the food. like you put sodium benzoate or all that stuff that's like near the end of the list on the ingredients. Those are the intention of those is to inhibit growth of bacteria.
Mhm.
So, why wouldn't they be doing that in the gut? So that's something that we're following up on right now. And then another possibility is that it's in the drinking water. I mean, there are lots of possibilities. It could be stress related, could be sleep, exercise related, could be just exposure, so we're following up on a number of things, but it could also be drinking water. Like the US drinking water and in all industrialized countries is fluoridated. So it has chloramines in it to kill bacteria, and it has fluoride, which is for your teeth and has great public health benefits, but it's also the purpose is to kill bacteria. So, maybe there's something else going on there.
It'd be interesting to know how like your economic standing changes too. Like if people who are, have less money, access to money, if their microbiome has less bacteria because maybe they don't have access to the food that like somebody who's has a couple millions of dollars or billions or something like that. See how that changes.
Yeah, we're looking at socioeconomic status a little bit in a study of infants with Children's Hospital of Philadelphia, and there's definitely some differences there. It's also always confounded with other factors, the access to certain diets. It tends to be confounded with ethnicity and race. It's confounded with where people live and how much green space is around. It's so it's been tricky to disentangle it with human studies, but I think it's probably also related.
Gotcha. Is it healthier to have more bacteria, or does it matter?
I didn't say that.
That's what I'm wondering. That's what I'm
right.
like. I don't like what does it mean to have more bacteria? Is that like
well, it's more diversity. So it's not necessarily a higher load.
Okay.
It's just more types. And generally speaking, more diverse ecosystems. Like if we look in the macro world, a more diverse forest is more stable to perturbation, to invasion by foreign species, and it tends to be it maximizes the biomass as well. This is something that was Dave Tillman's work. Who's like a really highly cited researcher in the ecology department, was that the biomass of an ecosystem is maximized when the ecosystem's diversity is maximized, biodiversity. So like from first principles, it should be better. From first principles too, like given that we co-evolved with microbes over the last millions of years, they are part of how our body works. And so you wouldn't think you'd want to grow up without some of them, but it doesn't mean that they're all good. And so that's still like a little bit of an open question, which ones are good, which ones are bad.
Yeah. Well, like, so if your example of like a forest, right, there's like a natural equilibrium of like forests for them to like plants to do niches in there. So if somebody has a less diverse microbiome, could potentially things mutate to fill in those niches that are missed?
Sure. Yeah, it might create more opportunity for opportunistic pathogens to emerge, for example, because it's just less full. All the niches are less full on average. So yeah, I think there are a lot of reasons why you would posit that it's bad to lose diversity, and we see that in basically every disease that's associated with a microbiome, there's a loss of diversity. The healthy subjects have higher diversity. So yeah, it's probably better to have a more diverse ecosystem, generally speaking.
Gotcha.
So Gabe found this, that this would give us like a 2x speed up. It was pretty easy. That's kind of cool. And then he had this idea that, every other optimization algorithm was optimizing one table at a time. Like, how can I make this search of query number 3 against reference number 99 faster? And he realized, wait a minute, we don't have just one query, we have 10 billion queries. So he had this idea that if we sort the queries alphabetically, and these are like just little random bits of genomes, they're going to overlap a lot. Like they're only 100 bases long. So the first query might start with TCGAA GGT, and then another 95 or 94 bases or whatever it is, 92. And then the next one might start with TCGAAGGA. That wouldn't be alphabetical, but so they're going to be the same for the first n bases, where that's determined by how broadly you're sampling the space of random strings. And the more total input queries you have, the more redundancy there is between one query and the next one alphabetically. And so he took this other approach where he said, all right, I'm just going to sort them all alphabetically. And then when I do one search, I'm going to cache the tables from all of the closest matches in the reference database for that search. And then when I get to the next query, I'm just going to walk along to the point at which it diverges from the previous one and hop into the table in that row. Because I already have the tables cached for the beginning of it. And when he did that, he found that that actually, on average, would prune the top third and sometimes more of a table. And so now you can see where this is going, because you can use this optimization to skip the bottom half. You can use this to skip sometimes the top half or more. And with some other tricks, you can keep track of, as you're searching through the database, this is more on this step. You can remember what was the best match you've already seen somewhere else in the database. So as you're working on the current reference, if you get to a point in the table where you can't beat an alignment you already found, you can just stop altogether. And combining these, he found that he was able to skip almost all of the search on average. So this is a tool that we call Burst. And the average, the worst case is still n times m, where you'd have to do the full alignment. But the average case across all of the queries is you're skipping almost all of it. And it was just kind of this, this cool synergy of these two optimizations, neither one of which really helps you out that much on its own, but they happen to optimize to completely, non-overlapping parts of the search space. And so with this tool, he could now do the same search in 5 minutes on the same supercomputer and get, guaranteed the best reference from the database every time. So what this meant is that we could now take less input data and get the same amount of signal out of it. Or, with a given amount of input data, we could get more signal out of it. And it's really exciting, kind of just from a theoretical perspective, to know that you can always get the optimal answer, when you're benchmarking tools and things like that. It's nice. It's like, driving the Ferrari instead of a moped. If you're kind of geeky about the methods part. So that's the first one, getting more signal from the data so we can do less sequencing. And then our colleagues, Kenny Beckman and Daryl Gohl at the Genomics Center, figured out how to miniaturize some of the steps to prepare the sequences for sampling. so that it could be more high throughput. And they got the costs there down quite a bit as well, to the point where we could do what we called shallow shotgun sequencing. And it was like just about the same cost as doing the 16S sequencing. And this allowed us to do it on big studies. And Ben Hillman, who's now visiting faculty at Macalester, he was another PhD student in the lab, and he did a lot of the analysis of, you know, showing that we could get a lot of the same information, not all of it, but a lot of it that you would get with deep shotgun sequencing using these optimal search methods and shallow shotgun sequencing. And so now I'm going to show you, well, first, any other questions about this before we move on?
How long did it take you guys to figure this out? Your explanation sounds like it's like, oh, they just did this in the afternoon.
It was pretty quick. It was like,
yeah,
it was like one day I stopped in and asked Gabe how it's going, and he said like, oh yeah, I implemented that thing, you know, with pruning, and like, it's cool. And then it was like the next week I asked him how it's going. He's like, well, I found something interesting, you know. And so it was not hard. I mean, the thing is, like, Gabe also, I mean, it might be worth looking at. Let's see. Might be worth looking at this. Like, he went in and it's all in like a single self-contained C file, and he just optimized the crap out of this. Like, he figured out, you know, certain instruction sets that he could use to parallelize parts of it using assembly. And so, you know, he figures out like what type of processor it is, and he's got these like custom binary masks that he put in, and this probably gave like another 10x speed up, you know, like, so the optimization gave 1000x maybe speed up on average, and then all these other things, you know, making sure that you're using, like, fitting within the cache lines, you know, so you're not crossing a cache boundary and whatever else like that, gave another 10x. So it's actually, it's documented, and enough that if you really like read through some of it, you can find out some of the things he did. And I think you could have like a whole graduate level course on this stuff. Like, to me, I think he's like half human. I can't do C like that. So yeah, it was pretty quick, but then he spent like another couple of years doing lots of other optimizations.
Okay, cool.
So now we can do shallow shotgun sequencing, and then we immediately ran it on this study where we ran it on like every single sample, and that's the study called just don't just read the label, that was run by Abby Johnson, who's now an assistant professor here at the University of Minnesota. She was a postdoc in my lab, and this was a study that was sponsored by General Mills, who's a local company, and really cool that they sponsored some kind of basic research like this, because it's not like we were, you know, evaluating a particular food technology. And in this study, it was just like this basic question. So we actually ran this as part of a class where everyone in the class well, they didn't have to be in the study, but they were allowed to if they wanted. They would collect a stool sample every day for a few weeks, and also record every single thing they ate every day for a few weeks. And then we did shotgun sequencing on all the samples. So it's hundreds of samples, and about 500. And then what we were able to do is get this high resolution look at what was going on at the species level in each person. And so because it's shotgun sequencing, you can get down to the species level. The other version, which is the cheap version, usually you only get down to like family or genus level. So not as specific. And this is the first time we were able to see species level variation in people over time with such a dense time series. So each block here is a person, and then the colors are the different species. And you can see all sorts of things just by kind of gazing at this. You might even feel like a little bit of a psychedelic effect. It reminds me of those, like, I don't know if you've seen those old, there was like these art things where you'd fill a bottle with colored sand. Kind of looks like that to me.
Totally.
And what are some of the things you can, like, what can you learn about the gut ecosystem by looking at this plot? Any thoughts?
There's a lot of that red at the top.
So there's a lot of this Bacteroides genus. Almost everyone has that.
Prevotella copri seems pretty rare, but a couple of them have pretty high or pretty wide bands of it.
Yeah, that's something that was a big deal like 5 years ago. Someone in the field realized, like, hey, some people have this bug and some people don't. Like, a lot of the other bugs are present in almost everyone, just in varying amounts, but this was sort of a binary thing. Like, you either carry this bug or you don't. So yeah, you're absolutely right. You're looking at this red right here. These three people have a ton of this bug and nobody else has any except for, these other three individuals have a little bit here. So it almost looks like a blood type.
In general, most of the people have a similar distribution.
Oh yeah, so a lot of the same bugs are showing up. Does anyone have exactly the same mixture as someone else?
No.
So the ratios are different. I mean, like, these two people look like twins, but this one has a lot more of this Bacteroides stercoris bug. There's always something different between any pair of people. Some people are a lot more different than others. Like, this person over here is kind of an outlier. This person is an outlier. These people in the middle have a lot more kind of neighbors. And you can make a tree out of these, and visualize them in that way. So you also can see, there's a lot of like kind of day-to-day variation, but people are like generally staying, they're maintaining the same average distribution throughout the course of the 2.5 weeks. And then of course we got, this is zooming in on Abby's microbiome. She gave me permission to show this. And she had like the most stable microbiome of anyone in the study. And she was very proud of that because she's a registered dietitian, in addition to having a PhD in nutrition. And this is all done, for basically because we've cut the cost of the shotgun sequencing down so much, it's about the same cost as doing a single time point of normal deep shotgun sequencing. And yet we get, all this temporal variation. And then we collected their diet. And so here's just the very highest level of diet. Like, orange is grain products. Pink is sugars, sweets, and beverages. So what can you learn about people's diets from this visualization?
What are those two columns in the middle that are just sugar?
Those it turns out what happened to them is they are computer scientists.
Just monster every day.
So these people chose for the course of the study to only consume Soylent, which is like a meal replacement beverage that's popular in tech circles. You know, the whole thing of like,
Such a weird name though.
well, it's reference to eating
Green one's the
people
best.
from an old movie, yes.
I know.
But they didn't want to have to, you know, decide what to eat, so you just eat Soylent all the time and wear the same thing every day, so you also don't have to decide what to wear. And so yeah, they surprised us with this and presented a very interesting kind of sub-study, because if you look at their microbiomes, they did not converge. So over the course of, this was about 2.5 weeks, they maintained their individuality completely. So even though they flipped their diet in a very extreme manner, their microbiome kind of like held its ground and the ecosystem stayed. So very extreme dietary interventions can't necessarily push the microbiome in 2 weeks into a new state.
What's the lifetime or life cycle of a biome?
Well, one bug bacterial cell can reproduce as quickly as 20 minutes in ideal growth conditions. You know, it could be a few hours in other cases. So it's pretty quick. You know, there's a lot of evolution happening from day to day inside you. Like, antibiotic resistance is coming up. You know, you take antibiotics, you're going to see an emergence of antibiotic resistance in the course of a few days.
Did you track any new species or anything that popped up? Like I know you got your graph here of the ones that were present throughout those days, but like if you had the new one pop up with the Soylent that wasn't there before.
You know, we might actually, we didn't look for just in these subjects whether there was anything new that they shared that they didn't share previously. That would be interesting and not too hard to check. The data for this is all up on GitHub as well. But yeah, no, we didn't look at that. I mean, the other thing is these two people were cohabitating. So in theory, they could be swapping microbes more easily, but they didn't. Maybe they did. Maybe there were a couple of minor members of the microbiome that we didn't look at. So, you know, we should check that. But overall, they stayed unique.
But they probably still drank water, right? So that's an interesting factor to consider.
Yeah, that's true. Probably the same water source too. And yeah, so that it's not like a very controlled study. You know, it'd be interesting to put people on this for a longer period, or like have people eat a more diverse yet nonetheless stable diet, you know, over time. But yeah, they kind of like, they made it really fun. And also we had to exclude them from some of the analyses because they had no dietary variation. But when you look at the other people, you see it's super spiky. Like we used to talk about how much the microbiome varies across time and across people. And now I had never seen diet data like this that was so high resolution, you know, temporal resolution, daily full dietary recall. And it's really spiky, really variable. And so, you know, the question was, how do you analyze this? Here's Abby's daily microbiome, here's Abby's daily diet. And some foods, you know, show up like this brownish color down here was eggs on the weekend. You know, some foods only show up with a cyclical temporal association. Other ones are just kind of by chance. You know, we're trying to figure out like, how do we analyze these data together, and how do we deal with this super variable dietary data? Maybe there's some space of latent functional data that we could, you know, in the diet features that we could like project them down to. Well, of course, there's nutrients. So that's the first thing we tried. And there's all the macronutrients, protein, fat, carbs, and whatnot that you get in on the nutrition label. And there's other micronutrients that are, tracked in the food database that we use. So this is what we started with, and this is what a lot of studies have done with nutritional data, is take the food list, which is very unique to each person, and project it down to nutrients, which are common across all foods. And in theory, you'd think that would work. But when we tried this, we found the nutrition labels didn't explain the microbiome variation across people at all. So this is a plot where, if you remember that original plot, here we're getting into a little bit more of like the data visualization side of things, but the original plot of the people from different immigration status groups, that was like showing, who's similar to whom, where the people still in Thailand were on one end and the US controls were on the other. This is the same type of plot where people with similar diets are close to each other on the plot. And those are the blue points. And then the red points are the same thing for microbiome. And then we did an analysis called a Procrustes analysis, where you basically rotate and stretch one until it aligns as best as possible with the other. And if these two feature sets are highly interrelated, then they would line up perfectly, because people eating similar foods or similar nutrients would also have similar microbes. But what we found is like it was kind of a hairball, and it didn't really work. And we tried this with lots of different distance metrics, different normalization techniques, using dry weight of the nutrients, using the moisture of the foods as one of the features. We could not get these to line up. And at first that was like a little bit, puzzling. But then we were thinking about this mystery food that Abby had shown me, that was had all these like crazy ingredients listed in it, and it looks like something from the worst processed food factory on earth. Look at all these different flavoring components, colors, preservatives, sugars, fatty acids, and so on. And you might not guess, this is actually a whole food that some of you might have consumed in the last week. It's actually a strawberry, and that's what's in the strawberry. It's sort of like, people get very chemophobic and talk about chemicals always being bad, but chemicals are just, anything occurring in nature. And these are all of the, chemicals that are in a strawberry. And so we realized, like, if we look at these ingredient lists in these nutrition tables, we're not even scratching the surface of what's actually in the foods. We're just tracking the 100 or so micro and macronutrients that we know are metabolized by the human body directly. But there's all this other stuff in everything we're eating that is metabolized potentially by your microbes. And when that happens, something can get broken down by one microbe, and then there's another conversion by another microbe, and so on. And this is whole like food network that's occurring in there. That's essentially like a little drug factory that's just constantly manufacturing byproducts that we know nothing about, and then we have no way of tracking. So we realized, well, of course the nutrition labels don't really correlate with the microbiome, because they're written for humans, not microbes. So then we said, okay, let's forget that idea of doing the nutrients. Let's just look at the foods and say, do people who eat similar foods also have similar microbial environments or microbial ecosystems? And here we were able to get, this was the only way we were able to get a significant like overlap between diet and microbes across people. And to do this, we had to go into some sneaky methods, like because the foods aren't all the same, we actually put the foods into a tree, a hierarchy, so that we could say, you know, you and I don't eat exactly the same foods, but they're more closely related than like you and this other person. And so then we can deal with, because the food matrix is really sparse because people eat unique foods, but we could sort of like use the tree to share statistical strength across related foods. So there's some computational stuff that happened behind this. But from this, you know, we could now see, okay, there's this cool stuff happening, you know, at the individual food level. And then we also saw that there were associations because we were able to do the shotgun sequencing on all the time series samples. We could actually ask for each individual person, do you have any associations between a particular food type and a particular microbe? So, you know, this is one plot of someone who their abundance of Roseburia inulinivorans is going up when they eat more cakes, cookies, bars. And some of these associations were held across multiple people. This is one of them. Roseburia inulinivorans, if you look at the name, what does that bug eat? Inulin, which is a very common fiber additive in like snack bars. Anytime you see a snack bar that says high fiber, if you turn it over, 95% of the time, it'll say inulin, parentheses, chicory root. And it's like this really small, easy to break down fiber that you can't really notice when you're eating it. They even put it in gummies. Like, how can you eat a gummy and get fiber? But it's because it's this one little fiber. And this bug, Roseburia inulinivorans, likes to eat it. And we saw this correlation across people. You know, when they eat more of these types of foods, they had more on a day-to-day basis of this bug. But most of the associations, the vast majority, were personalized. So we'd find things like Bacteroides oleciplenis was inversely correlated with pork consumption in some people and positively correlated in others. Bacteroides species 116 was, you know, again, inversely correlated in some and positively correlated in others with dark green vegetable consumption. And so there's a lot of unknown here still. It could be that people have slightly different strains of these species. It could be because of subtle differences in the types of pork or dark green vegetables they're eating. And it could also be that the individual species isn't the thing that matters entirely, it's the whole ecosystem. So, you know, maybe you and I both have this species of Bacteroides, but all the rest of my bugs are different from all of the rest of your bugs. And if we each eat dark green vegetables, you know, maybe in my gut, the Bacteroides, or in your gut, the Bacteroides species 116 is able to utilize them because of the combination of other bugs that they work with. And in mine, it actually enables some other bug that outcompetes it, you know, and so it could be that the ecosystem is sort of taking these foods down different metabolic routes. But the basic finding we saw was that these species diet associations were very personalized. You know, this is, as I said, like just kind of a arc of some things that we were doing in the lab, kind of like doing studies, seeing, you know, find something interesting, and then if there's issues with the data or we need other computational tools, then we'll go try to make them. And then once we have better computational tools, we might run a different study that you know, we know is going to be enabled by those computational tools. So it's a little bit of like a feedback mechanism. Like we're not just doing computational tools, but we're also not just running biology studies. We're kind of like doing whichever one makes sense at the time, and trying to have fun while doing it. And these are basically, you know, summary of like some of the things I showed. Like industrialization seems to be causing a loss of ancestral microbes. Maybe diet could help, but we've seen with these more higher resolution methods, diet microbiome interactions are very complex and personalized. So there's a lot more we need to learn there before we could use diet to like treat diseases or prevent diseases. And then I showed some new methods that make shotgun sequencing affordable so you can get high resolution data in studies. So I'm going to pause there and just see if you have more questions about the scientific work that I presented.
I was just wondering if you had some other things that you're going to use this set of data that you can look at to try to find correlations, or if you think you kind of got everything you could out of this set.
I'll tell you, this was a hard paper to write because we did so many analyses on it, and they were interesting. Like we did network analyses of which microbes seem to be interacting with which microbes in each person, and then which interactions were consistent, which microbe interactions were consistent across people. And you know, that after months of work, we just like removed it from the paper because the paper was too complicated. So there was a lot more that we did. And I would say, you know, one of the challenges was figuring out like, well, what's the most interesting, the most salient stuff that we're finding here, without getting too bogged down in, you know, in all of the little analyses that we did. So yeah, but there's probably other things like we didn't check that that suggestion of looking for individual low abundance species that popped up in the two subjects eating soylent. You know, I'm sure there's more stuff we can do.
Did the location of the bacteria matter?
Mm-hmm. The location, yeah. So that's something that is definitely relevant in some cases. Like there are diseases that tend to present in the small intestine versus the large intestine. You can have certain microbes that tend to hang out on cancer or in a tumor. And so there's a lot of work that's happening in kind of those site-specific analyses. It's challenging to carry out in humans because you basically need a biopsy. And it's hard to get those without like also disturbing the community. So you know, a lot of that work is happening, like Ron Bleckman at the University of Minnesota has done work on cancer microbiome associations using biopsies and also using cells that they culture in the lab and then expose to different microbes and see what happens to the genes that are being expressed and whatnot. So yeah, it matters and it's hard to do in humans.
From a computational standpoint, is it just from kind of prior experience, I suppose, is it more easier to find sort of pre-made solutions or code available on GitHub, for example, or is it more custom built for the specific research that's been happening?
I would say most of the basic analyses are there. Like doing the alignment, you know, problem that Burst solved, there's dozens and dozens of tools that do database search and alignment. So you could easily you could easily get them up and running, you know, in a couple hours. It's only when you need to do, you know, you kind of hit the boundary of insights you can gain from the data set. So like in this case, you know, Abby had to do a bunch of innovation to be able to compare your diet and my diet using the foods, because you and I probably don't eat exactly the same foods, like ever. And so she had to make a whole new tool for that. It's called Food Tree, because it projects all the foods into this hierarchy and then uses that to find the similarity of diets. And, you know, so that was like a whole thing. I would say, you know, that, I mean, it's hard to answer the question with a general answer, because it just depends. Like, it's 90% of the time you're just doing canned analyses where there's already a tool and you can pull it down and you don't have to do anything. But the other 10% of the time, you get to a really interesting, like, sort of edge case, and you have to make your own tool, and then you might spend a year doing that. So you might spend 90% of your time solving problems that only come up 10% of the time, 10% of the questions.
Yeah, and then as a consumer, I suppose, is all food the same? For example, a milk, do they kind of contain the same stuff at the same quantities, or is organic milk better with less things, or what have you guys noticed in terms of, right?
Well, yeah, we don't know from this. I mean, like, milk is fairly homogeneous, I think, you know, from milk to milk. But organic versus not, you know, our database, our food monitoring software did not collect organic versus non-organic. It also didn't collect, so I don't know. And we also didn't get anything about, there wasn't much about the food preparation. So, you know, if you take like spinach and you eat it raw versus steamed versus roasted or grilled, you know, you're probably changing the fiber, the shapes of the fiber substrates as you do that, which will change how the microbes react. Or like a banana is a great example I heard somebody use once. So like a ripe banana has different fibers in it from an unripe banana. So you can't just say banana. So it's a really hard area to work in, which is why Abby's doing that now. That's her work, is like, how do we better measure diet and deal with food to food variation and incorporate that into studies about human health.
Okay.
I'm curious about the category you had of egg. Is that like chicken egg or duck egg or?
Oh yeah, I mean, that was like, what was that category? That was, it was eggs.
Yeah. So
Yeah.
is that like just every eggs?
I think it was every eggs, but you know, probably for everyone in the study, it was chicken eggs.
Mm-hmm.
But is there a metric for that? So you said that you created a tree of the chemical components of the food. Is there if you're measuring similarity and differences, how do you say that, you know, one compound is more chemically different than another in meaningful ways? Is there a metric for that?
Yeah, I don't actually know. You got to talk to a biochemist, but
Okay.
yeah, like, so we weren't using the chemical, like the shapes of the chemical substrates in the tree. It was using a hierarchy of foods, basically like a food taxonomy that was already in the database. And so Abby was like trying to analyze these data and was looking at the database and realized it's already in a hierarchy. I just need to scrape this taxonomy out and put it into a tree structure. And then I can use the tree to project the highly sparse food data down to, you know, lower dimensional space. So it's basically like crowdsourced hierarchical labels of foods that were input by nutrition experts over the last 50 years into this database that's run by the USDA. It was not based on chemical composition, which is the right thing. That's like the future. It should be based on what are the actual substrates in the food, how are they affected by the preparation, and what's in common from food to food.
Very interesting.
So I'll talk just briefly also about this company that I started in case you have questions about that. So I did this with Kenny and Daryl. These are the guys that were helping to miniaturize those processes. And they had some other innovations for the marker gene data. And we both had been having like a lot of interest coming in externally for help analyzing samples and data that was more than we could handle that I could handle in my lab, or they could handle at the genomics center. And so we started this company that we called Core Biome, that was basically like a high throughput microbiome analysis lab that had its own sequencers, its own staff, and all of that implemented this. It actually was trying to sell cheaper data so that people could run big studies and get more signal than they would. So it was kind of a inverse of what some labs were doing, where they'd try to like charge as much as they could for the data. We were trying to drive the cost down so that people could run bigger and bigger studies. And it was over in located in the University Enterprise Labs, which is a privately owned like building with lots of little labs where lots of startups go. That's only like a couple of miles away. I happen to live over here, so for like the last 4 years of my life, I would like often make this trip, and it was doing half of one and half of the other, and then would also on good days stop at another stop on the way home, which is the Surly Brewing Company, where I actually went just last night with some of the original employees from the company. But so I got some experience with like that process, licensing from the university, trying to get investors to put money into the company, trying to build a sales process, the marketing process, and hiring people and negotiating. And we actually sold the company a couple of years ago, and then I've been, until recently, was still working there half time. So I went through that whole process. So if any of you have questions about this part of the translation of science into industry and sort of the commercialization aspects, I'm happy to talk about that, answer questions now, and you can also follow up with me separately.
Were you able to sell it to somebody who had the same ideals of keeping the costs lower?
Kind of. That's a good question. They didn't really have a choice, because we had already like put it out there and said, like this is for big data. We were trying to enable, that was like our business proposition, was enabling big data, and then also combining it with expertise on sort of the analysis side and dealing with tricky samples and whatnot. So they didn't really have a choice, because it was already out there. But it was tricky to, like, a lot of the investors wanted us to like, no, don't like commoditize this. Like, how, you need to be out there charging as much as you possibly can. But that just didn't make sense from a scientific perspective. So that was one of the things I found most challenging about it, was like trying to keep the company fiscally solvent, like meaning not let it go bankrupt, but also doing the right thing, given that constraint, doing as well as possible for the customers who were scientists, academic scientists and companies on the other end. And the university has a great technology commercialization office that's right here, that they know how to do all of this, all of the the legal parts. They can handle the licensing, dealing with other companies who might want to sponsor research and so on. They're a really great resource and gave us a ton of support. So yeah, again, feel free if you want to talk about anything specific about commercialization. Maybe you have ideas to do something, or you have other questions about the research or algorithms and whatnot. Feel free to reach out. I think I'm pretty easy to find and contact. And thanks again for your time, and for thanks to Kevin for inviting me here.
Thank you.
Yeah, thank you very much.
Thank you.
Thank you.
Thank you, Dan.
Thank you.
We're going to do, it's about 20 minutes. We're going to start the next talk. Dan will be around at about 9:45 to start the next talk. So take a little break. We'll come back in a little while.